So this talk has been constructed to really answer two commonly asked questions that are related yet distinct. And so the first question would be, why does it take so darn long for a new drug to come onto the market for Parkinson's disease? And then the second question is, why don't we yet have a therapy or a drug or a treatment that radically changes the way we treat Parkinson's disease, a disease-modifying agent that will slow down, stop, or actually even reverse the course of Parkinson's disease? So for the first question, I'm really going to talk about the clinical trials process. And this is really to give you an idea of the length of time it really takes to develop a drug from its discovery to approval. And then the second part of this talk, we're going to focus specifically on disease-modifying therapies. We've actually done numerous trials to look at a medication that might slow down the progression, but we've been unsuccessful. I'm going to explain why we've been unsuccessful, what are the lessons that have been learned, and hopefully give you an idea of what's in the pipeline. So what are clinical trials? This is just a definition so that we're all on the same page here. And this is taken directly from the NIH website. And so a clinical trial is a research study in which one or more human subjects, so these are human trials, this doesn't count trials that are done in animal models. So human subjects, they're prospectively assigned to one or more interventions. And so this is a treatment, a drug, or placebo, to evaluate the effects of that treatment or that intervention on health-related biomedical or behavioral outcomes. So how does that drug or intervention impact someone's function, someone's behavior, mood, whatever it is that you're trying to study? So it takes a long time to get a medication or a drug from development to approval. And we're talking a couple of decades here. And the drug development process is split into two sections. One is called the preclinical section, in which someone discovers a drug in a lab, and then it's tested out in animal models. And that may take several years. And then after that, it enters the clinical drug testing realm, in which a drug application is done, and then you go through 3 phases of clinical trials, and I'll spend some time talking about what's involved in those 3 phases. If it shows that it's effective, well tolerated, safe, the sponsor, the drug company, whoever has discovered it and tested it, can then apply to the Food and Drug Administration for a new drug application. It goes through a review process, which may take a few years, and then it gets approved, and then there's surveillance of that drug. Just to give you an example of some of the drugs that have been approved for Parkinson's disease and how long it takes, on the top you see Rasagiline, or Azilect is the brand name. It is an MAO-B inhibitor, and what that means is MAO-B is an enzyme that breaks down dopamine in the brain. So by inhibiting the enzyme, dopamine hangs around a little bit longer. That took about 15 years. And this is just clinical development. So this does not count preclinical or tests in rodent models, monkey models, etc. Requip, or ropinirole, took about 11 years for approval. And you can see in the various colors how long it took to go from a phase 1 to phase 2 to phase 3 type trial. And then at the bottom is something called glial-derived neurotrophic factor, or GDNF. This is a trophic factor that is present in the brain and encourages development of nerve cells. We've thought that placing or increasing those levels in the brain might help regenerate or preserve dopamine cells from dying out. As you can see, we're still having ongoing trials. There have been several trials that have not really shown significant improvement, and so we're still waiting. So let's talk about phase 1 trials first. What is this? So this is the very first time that a medication or a drug gets introduced into humans. So they've already been studied in animals, animal models. We think we know how the drug works, and we're going to try it in human subjects. And so as you can imagine, these are very closely monitored studies. Oftentimes they're done in a hospital setting where you stay a couple of days. And the goal here really is not to prove that it's effective. The goal here is to really study the safety profile and see whether or not people can tolerate the medication. So we're looking at the pharmacologic actions, and there may be a lot of blood draws to see what levels of how the levels of the drug are in patients over time, how the level of the drug is in other, like spinal fluid and urine, etc. You're looking at how high you can go before you get side effects, and you may get evidence on effectiveness, but that's not the point. So again, because it's the first introduction of a drug into a human, there are very small numbers in these studies, often 20 to 80 or so. Most often, it's healthy volunteers, and these are people who are paid to do this study. Rarely is it studied in the disease of interest. Most oftentimes healthy volunteers. Short trial duration, intensive monitoring, and again, because we're looking at safety, it's not expected that it would improve the health or the function in subjects. And based on that, if it looks to be relatively safe, relatively well tolerated in healthy volunteers, that's when you move on to the next phase. If it doesn't seem to be well tolerated at all, or it doesn't achieve levels in the blood in which you would expect it to be effective, you stop there and it doesn't go further. So an example of this would be this trial. This is a Coenzyme Q10 trial, phase 1 in Parkinson's disease. Now this is a little bit unusual because actually this phase 1 trial was not the very first trial of Coenzyme Q10 in humans. It was actually done after a phase 2 study. But the principle is we're looking at the safety in patients, and we're looking at how high of a dose can we give. These are Parkinson's patients, 17, so again, small number, given multiple doses of Coenzyme Q10, and baseline, and then 1,200, escalating doses, 1,800, 2,400. And really the point is, as you can see, after or above 2,400 milligrams, the level in the plasma or the blood does not go higher. So we know that going above 2,400 milligrams is not helpful, or it doesn't increase the effective number or amount that's in the bloodstream. So a phase 2 study is typically the first introduction of the drug into those with the disease. So again, phase 1, healthy volunteers, looking at safety. Phase 2 is the first one into those with the disease. So if you have Parkinson's and you are asked to participate in a research study, typically you would probably be asked to participate in a phase 2 or a phase 3 study. The goal of the phase 2 study really is to try and determine if the drug is promising enough to go into a phase 3 study, to further development. And really you're looking at further safety, further tolerability, and you're trying to get an idea as to whether or not this might be effective. So the goal is not to prove effectiveness. It's really to prove, again, safety and tolerability in patients with the disease of interest, and hopefully getting some efficacy data. All right. So these are typically larger studies than your phase 1 studies, so hundreds of subjects. Again, these are all well-controlled, closely monitored. This is also where you might start to have a placebo group, which is an arm in which you give a pill or an identical treatment that does not have any active study drug in it. You might explore a wide range of doses to see which one might be more effective. And because it's the first in the disease, in patients with the disease of interest, it may improve some disease symptoms. So going along with this coenzyme Q10 pattern here, this is a phase 2 study in about 80 subjects or so with for coenzyme Q10. And they enrolled patients with Parkinson's disease who had not been treated with any medications, so levodopa, dopamine medication naive, followed them for 16 months. And again, you're just trying to see, they started with 300, 600, and 1200 milligram arms. And what you can see is really at the end, the placebo group, in here are the 300 and 600 milligram groups. They kind of are not that much different than placebo. And you have this 1200 milligram group, which looks like it's separate from placebo. And what we're testing here is the UPDRS. It's the Unified Parkinson's Disease Rating Scale. It is a measure of how severe someone's Parkinson's disease is. And you can see that after 16 months, these are patients, again, who have not been treated. The scores seem to worsen a lot more in placebo than in a 1200 milligram group. But this also brought up the question, so if 1200 milligrams has this separation, what about higher doses? And that's why they ended up doing that phase 1 study that I showed you earlier to see if people could tolerate higher doses than 1200 milligrams. Eventually, based on these this study and the phase 1 study, they then tested coenzyme Q10 in a phase 3 study looking at placebo compared to 1200 milligrams compared to 2400 milligrams. Unfortunately, that did not turn out to be a disease-modifying therapy. There was no difference. But this kind of goes to show you how these medications or these drugs progress in the pipeline. So phase 3 studies are the most important in terms of this is the study that the FDA will use to approve a drug for the market. And so it's comparing the investigational drug to a placebo arm. Or sometimes, if it's dangerous to put someone on a placebo, the condition is just so severe that you can't justify putting someone on no treatment, they might compare it then to the best known treatment. And you're really trying to prove, is this new drug better than the placebo, than the other option? Are you improving things beyond that? And so it's going to include hundreds of patients with the disease. These are double-blind, typically randomized studies, and placebo-controlled. So double-blind means that if you enroll in the study as a subject, you don't know what treatment you're on. The physician who's in charge of the study, the team that's in charge of you, do not know what treatment you're on. And then you're randomized. There's a process in which you might be put on drug, you might be put on placebo, no one knows. Study population is clearly defined. What that means is they're looking for patients, so in this case Parkinson's disease, but they'll have all sorts of requirements of what you need. So you have to be within a certain stage of Parkinson's, you might have to have certain symptoms, you can't have other symptoms. So it's a very well-defined population, and basically the purpose is to kind of decrease all of the variables that might confound or confuse interpretation of the results. Again, involves a lot of exams, and really this is the one that decides whether or not this drug is going to be approved for use. And rather than show you the coenzyme Q10 study, which I already told you was not a positive study, I decided to put this one instead as an example. This is CVT-301. That was the code name for what is now Inbrija, or the inhaled levodopa. And so this is a phase 3 study. As you can see, people were randomized to 3 different arms, a 60 milligram dose, an 84 milligram dose, and a placebo, followed for 12 weeks, stopped, and then they could have continued on in an open label extension. And what they were looking at is, so at baseline, they were trained and then over time followed, and then really the outcome measure here is at 12 weeks. When you brought in someone who was off medications, you gave them the inhaled levodopa dose, at 60 minutes after they got their dose, what was the change in the UPDRS? Again, this test of motor severity. And what you can see is that after about 10 minutes, this medication kind of seems to take effect in that the UPDRS score improves over the first 10 minutes to half hour. After that, seems to be about the same, and they only followed patients up to about an hour afterwards. The 60 and 84 milligram groups were pretty similar. The 84 milligram group was a little bit better than placebo here. And here's another way of looking at it. Almost 60%, 55% of patients on inhaled levodopa had basically turned on at about an hour after their dose, compared to only 36% of the placebo. So it was based on this trial showing effectiveness of the inhaled levodopa that the company that produced it applied to the Food and Drug Administration, or the FDA, for approval. It went through a couple of rounds of review and then was finally approved earlier this year. Now, after a drug is approved, that doesn't mean all monitoring stops. There's what's called a phase 4 study in which patients can be monitored, and you're really looking, or at least the companies that kind of do these types of things are looking at post-marketing, continued safety parameters, might compare it to other similar drugs on the market to see how it's doing. But again, this is where sometimes you find out that a medication might have some safety risks, cardiac risks that you didn't find out in the trials just because the trials were shorter, or they might have involved fewer patients, and it's not until more people get on the medication or followed for a longer period of time that some of these other side effects pop up. And so that's why you might hear of drug recalls later on. All right, moving on. So that's why it takes a long time to get a drug approved. And so the question is, when potentially are we going to get a disease-modifying drug for Parkinson's disease? What is disease-modifying? I've kind of alluded to it earlier, but it's really a treatment intervention that either slows down, stops, or even reverses disease progression. So this is what we all want, right? This is as close to kind of a cure as we can get it. Something that slows it down is not a cure, but if it can stop it and actually reverse it, that would be. Unfortunately, right now, to date, there has been no agent that has been definitively proven to have a disease-modifying effect. And I'm going to spend some time talking about the different trials that have gone on that have failed, but to give you an idea of how difficult it is to design a disease-modifying trial, but also give you an idea of what lessons we have learned from the past. Before we do that, there are some obstacles to actually conducting some of these trials. So a disease-modifying trial, if you're looking to slow down the progression of the disease, the ideal patient population in Parkinson's would be someone who's early on, right? Because if you're going to slow it down, someone who's a little bit more advanced you might not be able to see as much of a difference as someone who's early on. But that is a population in which it's hard to recruit. Oftentimes, you're just diagnosed. You're dealing with the diagnosis. You don't know what your eventual course is going to be. There are all sorts of questions that need to get answered and settled in your mind before you might even think about participating in a trial. So there's usually a lack of willing participants for these early trials. And with these disease-modifying trials, you need to be put on placebo, or it needs to be tested with a placebo. And of course, if you have Parkinson's disease, it's unlikely that you want to take that chance to be put on a placebo drug. And then disease-modifying trials are just very long, right? If you're looking to slow down clinical progression, you have to follow your course long enough to be able to tell whether there's a difference in the course that you're going to take. The longer the trial, the more it costs, the more effort it takes to kind of keep people in trials. And then, of course, there's that inconvenience. 3 years, 5 years of just going and getting study visits can be burdensome. And then involving just hundreds and hundreds of patients, you have to have multiple sites, and then it gets hard to kind of standardize operations. Even before that, though, even before a drug gets into the testing phase, it's sometimes hard to identify a good agent that you think might change the course of the disease. Partly because we're still not exactly sure what causes cell death in Parkinson's disease. We don't know exactly why the dopamine cells kind of die off. There are a lot of theories, and you might hear things like oxidative stress or inflammation or all these kind of processes that go on to cause cells to kind of deteriorate over time. But what we don't know is if there's one thing that triggers everything else, do they occur at the same time? Is one type of process compensatory, meaning one thing goes and causes deterioration, the other thing happens to try and protect it from deteriorating? We just don't know. So it's hard to identify one specific silver bullet that's going to stop progression. These are also tested in animal models first, which is important to make sure that it's going to be effective, but the animal models don't really mimic what happens in humans. So the way we develop animal models is usually you have a healthy mouse or whatever the animal model is, you give them a toxin to produce Parkinson's. That's not really generally how it works in humans. So are we really developing a drug to do or to slow down what happens in humans by testing them in these types of animal models? It's hard to know what dose you need when you're testing animals, and then how does that translate into humans? And then really I think the main thing, the thing that I want to go over is what are the endpoints? So what are we treating and how the endpoints that we've chosen for these trials have been difficult because of symptomatic effects. And I'll explain that more clearly in a little bit. So clinical endpoint in a trial is a characteristic or a variable that reflects how a patient feels, functions, or survives. What that means is if you put someone on a drug for a trial, you want to know that if you're, for example, going to slow down the progression of disease, you want to pick something that reflects that slowing down. So what are you going to do? You might say, let's see how patients are functioning in terms of their motor symptoms. And so we've chosen this UPDRS, this rating scale that kind of gives a sense of motor severity. And that's your clinical endpoint. It might be different for different disorders. This slide is in your handout to kind of give you an idea of different endpoints. I'm not going to go over it. Instead, I'm going to use some of the trials that I've put up here to illustrate how these endpoints are used. So the very first study that really looked at a drug that might slow down diseases is the DATATOP study. So this was done in the late 1980s, published in 1993. And what it did was looked at Surrogate, which we know is an MAO-B inhibitor. Again, MAO-B breaks down dopamine, and so this allows dopamine to hang around a little bit longer. At the time the trial was done, we did not think that it actually had any symptomatic effect on Parkinson's disease. And so what do I mean by symptomatic effect? If you have Parkinson's, you oftentimes get placed on a medication like levodopa. You take levodopa, you feel better, you move better, the tremor is better. It's treating the symptoms, and so it has a symptomatic effect. What you ideally want when you're testing a drug that slows down the progression is to not have these symptomatic effects, or to have an endpoint that doesn't use these kinds of symptomatic effects. Meaning, if I give you Surrogate, what we know now is that it actually improves the symptoms. So I give this medication to you and then rate your motor symptoms, you would actually notice an improvement after taking Surrogate. We didn't think this back then. And so you have 4 arms. This is Surrogate, which was known as Deprenyl back then, and then tocopherol, which is vitamin E. And there are 4 arms: placebo, just vitamin E, just Surrogate, and then a combination of Surrogate and vitamin E. And what they decided to do was they decided to use an endpoint of how long it takes to need levodopa. So you enroll patients who are not on levodopa, and then the longer it takes you to need or require levodopa, you're slowing things down, right? And so what they saw was those patients who were on Surrogate, it took 719 days after enrollment in the study to need levodopa, whereas if you weren't taking Surrogate, 454 days. So this was touted as a great trial. We have a medication that can slow down the course of Parkinson's disease, but again, then we realized that selegiline has a symptomatic effect. It's like giving levodopa and saying, or equivalent like levodopa and saying, oh, it takes longer, or it takes longer to really need levodopa. You're already treating the symptoms, so you don't need another medication. So how do you then separate these symptomatic effects from the disease-modifying effects? And so some smart people came up with what's called the delayed start design. And so this is how this works. You have a group and you give them the medication or the study drug immediately. Right? So that's called the early start group. The other half of the cohort gets a placebo. But at some time afterwards, 9 months, a year, that placebo group then gets started on the medication. And so if you're just treating the symptoms of Parkinson's disease, it shouldn't matter if you get it early or later. The delayed start group should basically catch up to the first group. And that's just a symptomatic effect. But if it makes a difference, if it's a disease-modifying effect, the earlier you treat someone, the better off you are. And so then the delayed start group, it would be like this. There might be some symptom improvement, but it would never, ever catch up to the first group. So this is, this design is used, has been used in a couple of different trials in Parkinson's disease. But it has been problematic in interpreting some of these trials. The most well-known is called the ADAGIO trial. The ADAGIO trial looked at Rasagiline, which is another, which is Azilect, another MAOB inhibitor similar to selegiline. And we're looking at MAOB inhibitors because in these animal models, it really looks like it's very protective of dopamine nerve cells, which is why there are multiple studies of these MAOB inhibitors. But again, so patients were randomized to a 1 milligram dose of Rasagiline or a 2 milligram dose or placebo. And then about 9 months later, the placebo group gets started on 1 milligram or 2 milligrams of Rasagiline. And the point here is, so this is the 1 milligram a day group. They start on the early. The delayed start, they improve a little bit, but they never catch up to the first group after 72 weeks. So maybe this is a disease-modifying effect. But then you look at the 2 milligram group. Okay, they start, the delayed start group starts, they have an improvement, but by the end of the trial, they actually catch up to the first group. So how do we interpret this trial? So some physicians, clinicians, have interpreted this as, well, you know, 1 milligram shows that doesn't catch up, that's maybe disease modifying, why not start it and put patients on it? Another way to look at it is, well, Rasagiline 1 milligram is not strong enough to cause a catch-up, and if you give 2 milligrams, it's a stronger dose, you're going to catch up, so Rasagiline is not disease-modifying, and that's where you have a lot of controversy. So you, how many of you actually take Rasagiline? So some people do, and so some people, you might have a physician who's kind of the, well, this data shows that it slows things down, might as well be on it. The rest of you might not have, you might have a physician that says, well, this is not clear. You might be exposed to side effects. Rasagiline does have drug interactions. It's not worth it because we don't think it's disease modifying. So what other ways have we come up with to try and test a disease modifying effect? Number of years ago, the NIH came up with this group, NET-PD, which is Neuroexploratory Trials in Parkinson's Disease. And their theory was this. If you're going to change the course of disease in Parkinson's, it's going to be obvious, right? If you find a home run, it's going to be clear. It doesn't matter what medications you give. This whole symptomatic effect doesn't really matter. You give someone a drug, if it modifies the course of the disease, you're going to see it. So what they decided to do is this futility design and then followed by a large, simple study. The futility design was designed to screen a whole bunch of drugs and find the drug that has the highest signal, the one that's most likely to be a home run. And then you take that and then you take a whole group of patients, half of them get the study drug, half of them get placebo, follow them for 5 years. Doesn't matter what you do with them because if it's going to modify the disease, you're going to see it at the end of 5 years. So that's the theory behind this. And so the first, I'm sorry that this doesn't show up well, but the first futility studies looked at 4 agents, Coenzyme Q10 was one of them. There was this compound called GPI 1485, creatine, and minocycline. And of the four, creatine showed the highest signal as most likely to have a disease-modifying effect. And then they took creatine, which is actually a dietary supplement that we use to improve performance in athletes. They then tested creatine in a large, simple study. So 1,741 patients, a lot of patients, followed for 5 years. And they used this primary outcome measure, which was it's a global statistical test, but it was a combination of different endpoints. So they looked at some motor endpoints, cognitive endpoints, gait problems, and quality of life, thinking that if you're going to make a difference in the disease, you're going to impact all of these different things. Halfway through, so 955 patients completed the 5 years. The other half had not yet. They decided to do an interim analysis just to see what the data was like, and it showed that there was no difference between the two groups. So they halted the trial prematurely and said, well, this is not making that huge difference. So again, another failed trial for disease modification. Unfortunately, the bad news kind of continues. Recently, basically within the last year, there have been a couple of trials looking at some disease-modifying agents that have not worked out. First of these was steady PD3. This is isradipine. Some of you may have heard of this. Isradipine is a calcium channel blocker, and it's used for high blood pressure. And there's some basic science data showing that it might help protect dopamine nerve cells. And so there was a phase 3 trial looking at over 300 patients with early Parkinson's disease, not really treated with anything, although some of them may have been on an MAOB inhibitor like Rasagiline or Rasagiline. And again, two groups, isradipine, placebo, followed for 36 months. The trial was completed, but at the end of the trial, there's no difference between the groups at 36 months. So this, again, not a disease-modifying agent. And then this trial, SurePD3, looking at urate. So it's actually, urate is a chemical that's in the blood, and if it's elevated too much, it causes gout. But there were some epidemiologic studies, some clinical studies showing that those patients who had higher levels of urate had a slower progression of their Parkinson's disease. So the theory behind this trial was, well, let's take patients who have low levels of urate, give them inosine, which is a precursor to urate, so it gets changed into urate in the body, and let's elevate their urate levels and see if we can slow down progression. And this was reviewed in late 2018. There was an interim analysis, again, one of, halfway through, half the patients had completed it. Let's take a look and see if there's any signal. There was absolutely none, so they basically stopped it and were closing out of this study. So then, if we're not sure if clinical endpoints are effective, maybe we can do some things like biomarkers. So biomarker would be something that you can measure and evaluate that kind of parallels the clinical endpoint. And so an idea would be a blood level. If you could follow blood levels or chemical in the spinal fluid, as the disease worsens, it gets larger, or you can detect it more. If there's no disease, it kind of falls. You could do it with imaging. Maybe if you image the brain, maybe as disease gets worse, size of the brain gets smaller, maybe there's something that you can do. So some trials have kind of looked at biomarkers. And so this ELLDOPA study basically gave patients, randomized patients to placebo, multiple different levels of levodopa. This was done to try and see if this hastens the progression of Parkinson's disease. And what you can see here is these patients were followed for just a little bit less than a year. This is the placebo group, and all of the patients that were on levodopa at the end, so they reached an endpoint, they came off the drug for 2 weeks, and then they were evaluated. And all of the patients on levodopa were much better off than those patients on placebo, suggesting that levodopa doesn't hasten disease progression. It might be modifying disease progression, slowing things down. Then they had an imaging arm of the study. So the image that they decided to use was a SPECT tracer. It was a dopamine transporter binding agent. So this is an agent that gets injected in the veins, goes to the brain, binds to dopamine transporters in the brain. You may have heard of DAT scan. This is kind of similar to that. And the thought is that if you have more dopamine transporters, there's more dopamine nerve cells in the brain, and you haven't progressed quite as much. If you have less dopamine transporter uptake, there's less dopamine nerve cells, and your disease has progressed. And unfortunately, in this trial, the imaging studies showed less uptake at the end of the trial, which would suggest that the disease had progressed, conflicting with the clinical results. So again, we're not quite sure how to interpret this. It probably is that the biomarker, meaning the imaging study, is not that accurate, and it's not a biomarker that we can rely on to suggest clinical progression. But I don't want to leave you with no hope or little hope, seeing as how we've gone through all of this without any improvement in finding a disease-modifying agent. Even though all these trials have been negative, I still think it's been worthwhile, and we've learned a lot of valuable lessons. And the valuable lessons are that it's very hard to select a clinical endpoint, and we really need to try and find more biomarkers, something that will allow us to detect disease progression without relying on how someone is doing, because medications can confuse, or it's hard for us to interpret how patients are doing just on the basis of motor, because there are medications that can improve your motor symptoms. So there's something called the Parkinson's Progression Markers Initiative, or the PPMI, sponsored by Michael J. Fox Foundation and conducted by the Parkinson's Foundation. And what they're doing is they're enrolling a bunch of patients, or a bunch of people with Parkinson's disease, de novo, meaning they have not been treated with levodopa, following them over many years, and they're just getting a lot of information on them. So sampling, getting their blood, spinal fluid, urine, DNA, getting all sorts of imaging assessments, all sorts of clinical assessments, from motor scores to behavioral, mood scores, cognitive scores, with the real hopes of finding one of these biomarkers, something that will allow us to reliably predict progression in Parkinson's disease. And then there are a whole bunch of other medications, agents that are being tested now. This is the last slide in your packet, and I actually changed this a little bit. The difference is the slide in your packet, I went to clinicaltrials.gov. That's a website that anyone can go to, and you can search for ongoing clinical trials in Parkinson's disease. So actually you can search it for any disease, but if you put in Parkinson's disease, then you can narrow it down to whether it's the US or Europe, but it's a website that you can search for these types of trials. And I listed the current trials that I could find of disease-modifying therapies in Parkinson's disease. But then I realized it's really not a comprehensive list. There are a lot of other trials that are ongoing, and so I changed it to this, which kind of gives you more of a sense of the different classes of medications. So we're not going back to the MAOB inhibitors and retrying that and trying to prove that it slows things down. These are new classes of medications that we think might be able to slow down progression in Parkinson's disease. The first one is the GLP-1 agonist. So this is, it's called glucagon-like peptide 1. It's actually used for treatment in diabetes, and in animal models of Parkinson's disease has been shown to be neuroprotective. The one that's gotten a lot of press is exenatide. I don't know if any of you have heard of that. It recently got a lot of press because it was tested in the UK and maybe thought to slow down clinical progression of Parkinson's disease by a very small amount if you look at the UPDRS. And I know there are multiple other exenatide-like medications that are being tested or that are going to be tested fairly soon in the United States. There's a class of medications called the c-Abl inhibitors. c-Abl is a tyrosine kinase inhibitor that is actually used for, so some of these c-Abl type inhibitors are used or approved for treatment of various types of cancers. And again, they also have been shown to potentially slow down progression in Parkinson's disease or preserve dopamine nerve function, and some of them are being treated. The one that has gotten a lot of press is called nilotinib. You may have heard it. It's used to treat leukemia, and there was a report presented at a meeting that showed that people with Parkinson's or Parkinson's dementia reversed or improved significantly, and so it's now being tried. But that was a case series, meaning that there was no placebo control. It just kind of observed people, and so it's being tried in a placebo-controlled fashion now. There are actually antibodies to alpha-synuclein that are being tested as well. So alpha-synuclein is a protein that accumulates in the brain of someone with Parkinson's disease. It's what's in the Lewy body, if you've heard of the Lewy bodies that kind of go in the substantia nigra. And there's a theory that alpha-synuclein toxicity, this extra alpha-synuclein that contributes to the degeneration in Parkinson's disease. So if you give antibodies to that, maybe you can improve things. It's a large number of antibodies are being tested, sponsored by different companies. And then the last is iron chelation. So the theory is that there is some iron overload or increase in the substantia nigra. This kind of causes increased stress in the brain and leads to deterioration. And so by chelating the iron, you kind of bind the iron and take it out. Maybe that can help slow down the progression. And deferoprone is one of the names that that's used, and you might hear about that in the near future. So we still have a lot of stakes in this. There are a lot of possibilities, and I hope that within the next decade or so, we'll have something that really slows down the progression of Parkinson's disease and can change the way we treat everyone and improve everyone's quality of life. So that's what I have, and I'm happy to take any questions from you. Thank you.